Hello friends, today in this video I am going to discuss about the restriction and modification system. Okay, so at first I discuss about the restriction enzyme. That means what is restriction enzyme? Restriction enzyme is one type of enzyme which restrict or inactivated the foreign DNA entered into the bacterial cell by, for example, from the virus, that is the viral infection. Okay, so the function of the restriction enzyme to restrict or inactivate those foreign DNA, that means those viral DNA, by cleaving or cutting those foreign DNA. Okay, so now suppose this is a bacterial cell. This is a bacterial cell, okay, and its genomic DNA, that is its own DNA, is present here like this. Okay, this is the genomic DNA of this bacteria. Genomic DNA of this bacteria, and here a foreign DNA entered into this bacterial cell from the viral infection, from the virus. For example, from the virus, this viral DNA entered into the bacterial cell, and now the restriction enzymes present in this bacterial cell recognize particular sequence in the foreign DNA and cut or cleave this foreign DNA like this and make small fragments of the foreign DNA. Okay, so now the question is how these restriction enzymes recognize this foreign DNA and how they cut this foreign DNA. Okay, so now at first we have to discuss about the restriction enzyme. So now the restriction enzyme that I denoted here as a RE is also known as the restriction endonuclease. Is restriction endonuclease enzyme. So here you can see that this restriction endonuclease is a nuclease type of enzyme. That means the nuclease enzyme cut the DNA in the phosphodiester bond in between two nucleotides. So suppose this is a DNA. Okay, this is a DNA, and here a nucleotide is present. Nucleotide number 1 and nucleotide number 2 is present, and this two nucleotide is connected to each other by the phosphodiester bond. So this bond is called the phosphodiester bond. Okay, and the nuclease enzyme cut this phosphodiester bond between two nucleotides and they are separated from each other. So in this way the nuclease enzyme can function. Okay, so now you can here see that a endo term is present. So actually the nuclease enzymes are two types. One is exonuclease and another is endonuclease. Okay, so at first we discuss about the exonuclease. The exonuclease enzyme start cleaving the DNA molecules from one end. That means they can cut the DNA, start to cut the DNA from either this end or either from this end. So suppose here a nucleotide 1 is present, and nucleotide 2 and 3 present, and this exonuclease enzyme at first cut the phosphodiester bond in between the 1 and 2 nucleotides. So now the first nucleotide is separated, and then they cut the phosphodiester bond in between the 2 and 3. So in this way they can cut the whole DNA. Okay, so but remember that exonuclease enzyme cannot cut the DNA in between any position of the DNA. They only can cut the DNA from the extreme end of either side. Okay. So now, what is the endonuclease? What is the endonuclease enzyme? This endonuclease enzyme is the opposite of this exonuclease. That means this endonuclease enzyme can cut the DNA in any position of the middle of this DNA, but they cannot cut the DNA from the end position. Okay, so as the restriction enzyme is the one type of endonuclease enzyme, they can cut the DNA in between any position of this DNA. But remember that there has a regulation in the restriction enzyme. That is, this restriction enzyme particularly recognize a particular sequence. That means suppose here a sequence is present like GATC. A sequence, this sequence is present, and a restriction enzyme particularly recognize this sequence in this DNA strand, and then they cut in any particular position of this basis. Okay, so this is the function of restriction enzyme. That is, they act as a endonuclease enzyme, but they recognize a particular sequence to cut the DNA strand in the phosphodiester bond in between two nucleotides. Okay, so in this way restriction enzyme function. And now the most important question is how this restriction enzyme distinguish the foreign DNA from the genomic DNA. That means how they distinguish that, differentiate that they have to only cut the foreign DNA, but they do not cut the genomic DNA of the bacteria. They only have to cut the foreign DNA. Okay, so how they distinguish this foreign DNA from the genomic DNA? Okay, so this is done by the modification enzyme. This is another type of enzyme which modify the genomic DNA of the bacteria. Okay, so let's talk about the modification enzyme. Okay, and this modification enzyme is also called the methylation enzyme. This modification enzyme is also called the methylation enzyme. Modification enzyme is methylation enzyme. Okay, so the name suggests that this enzyme modify the DNA by methylating the DNA strand. Okay, so how they methylate? Suppose this is a DNA strand, and this modification enzyme recognize a particular sequence in the DNA strand. Suppose here also a sequence is present, that is GATC. Okay, and this modification enzyme recognize this particular sequence, and they modify this sequence by adding the methyl group, that is the CH3 group, only in the adenine base or cytosine base. So here they only modify the base by adding only in the adenine base or in the cytosine base. That is, they can either methylate the adenine base or the cytosine base, but not the any other base in the DNA strand. So in this way, now the DNA is modified, and now when the restriction enzyme come to cut this DNA, they are prevented by this modification, so they cannot cut this genomic DNA. But here in the foreign viral DNA, there is no modification is present. So this restriction enzyme come and recognize a particular sequence of 6 to 8 base pair, and then cut this foreign DNA in those particular sequence. Okay, so in this way restriction enzyme and modification enzyme can function, can function. Okay, so Now remember that the modification enzyme can be conjugated with the restriction enzyme and they can function togetherly, or the modification enzyme can function independently. Okay. Now I discuss about the gene for restriction modification system. Okay. So in E. coli strain K12, at first the restriction modification system is discovered, where 3 genes are present which are involved in restriction modification system. Okay, and these 3 genes are called the HSD gene, that is the host specific defense or host specificity determinant. Okay, and these 3 genes are HSDS, HSDM and HSDR. At first, the HSDS gene products, that means the proteins produced by this HSDS gene, helps in recognition, helps to recognize the particular DNA sequence. So suppose this is a DNA and the HSD gene product particularly recognize a DNA sequence in the DNA, and this particular DNA sequence is called the recognition site or restriction site. Restriction site. Okay. So this is the restriction site, and the second gene, that is the HSDM gene. This HSDM gene product helps in modification. That means they modify the DNA by adding the methyl group. That means it is a methylation process, and that's why this type of enzymes are called the methyl transferase enzyme. Okay, that means they transfer the methyl group to this restriction site in any base. Okay, in adenine or cytosine base. Okay, so now the HSDR gene. HSDR gene product helps in restriction. That means they restrict or they cut or cleave the DNA in the restriction site. Okay, so in whole, that is at first the HSDS gene recognize the restriction site. Then if it is a genomic DNA, then the modification system modify the DNA by adding the methyl group. Okay, and in this case the restriction enzyme do not show their function, and if it is a foreign DNA, then restriction enzyme then the HSDS gene product at first recognize a particular sequence, that is the restriction site, and then the restriction enzyme come and cut this DNA in the restriction site. In this case, for foreign DNA, the modification system do not show any function. Okay. Now I discussed about the gene for only modification enzyme system. That means this type of modification enzyme systems only modify the DNA by methylation, but they don't have the restriction property. Okay, so at first I discussed about the DAM gene. Okay, this DAM gene DAM gene product is the DNA adenine methyl transferase. That is, this enzyme is a methyl transferase enzyme, and this enzyme recognize the sequence, that is the G8TC sequence, and then after recognition they add a methyl group in the adenine, and in this way they modify the DNA. Okay, and the next is the DCM gene. This DCM gene for modification is known as DNA cytosine methyl transferase. So it is also a methyl transferase enzyme, and this enzyme recognize this particular sequence, that is CCA or TGG. Okay, and then after recognition they add a CH3 group in the internal cytosine residue. Okay, so the last gene system for modification, that is the MCR system, and this modification system is different from this two modification system. Okay, and this MCR system contain two genes for this modification, that is the MCR B and MCR C, and these two gene product, that is the enzyme product, recognize a particular sequence which are already methylated. Remember that they are not methylated, they are modified they modified this sequence, but here this is already modified DNA sequence, and then this MCR system gene product recognize this modified DNA and cut this DNA sequence in the cytosine residue. Okay, so this MCR system recognize the A or G, then C, and then N, that is any nucleotide, and then A or G and C. So they recognize this particular modified sequence where the modification is present in the cytosine residue, and then they cut the DNA in the cytosine residue. So the main thing is that in DAM methylation and in DCM methylation, the modification is done by these enzymes. Okay, but here in MCR system, the modified DNA is cut by this MCR B and MCR C gene products. Okay, now the question is when the restriction modification system works. Okay, so in case of genomic DNA or internal DNA, the DNA can present in two forms, that is the fully methylated form or in hemimethylated form. In fully methylated form, that means in both DNA strand the methylation is occurred, the methylation is present. Okay, so suppose this is a DNA duplex and methylation is present in both DNA strand. Okay, so now when the restriction enzyme come and encounter that both the DNA strand is modified, so they cannot work. So here in this case, in fully methylated form, the restriction enzyme do not work. Okay, so now the modification enzyme come and encounter that already the both strand are fully methylated, so they do not have to work anything, do not have to modify anything. Okay, so that's why the modification enzyme also do not work here. And now when this fully methylated DNA replicate, they will produce hemimethylated DNA strand. That means one strand is methylated and another strand is non-methylated because it is newly produced DNA strand. Okay, so suppose this is the old strand where the methylation is present. Okay, oh sorry, and the new strands, there is a new strand where no methylation is occurred. Okay, and now when the restriction enzyme come, and the properties of restriction enzyme is that if there is any modification present in any of the strand, then they cannot cleave those DNA, and as it is, and that's why when the restriction enzyme come in the hemimethylated strand, then restriction enzyme encountered that the one strand is methylated, one DNA strand is methylated, so they cannot cleave the DNA. Okay, so in hemimethylated strand the restriction enzyme do not work. Okay, and the modification enzyme come and encountered that one strand is methylated and another strand is non-methylated. And remember that the methylating enzyme only work when the DNA strand is hemimethylated. Okay, so now here DNA strand is hemimethylated and the modification enzyme come and methylate the other DNA strand like this. Okay, so now these two strands are also fully methylated. Okay, so now in case of foreign DNA or external DNA which is non-methylated, so let me draw. This is the foreign DNA. Okay, there is no methylation present. Now restriction enzyme come and encountered that there is no methylation present in this DNA. So they recognize the particular sequence and cut the DNA like this in particular DNA sequence. Okay, so here restriction enzyme work. Oh sorry, I write here modification enzyme work. Okay, so now here the modification enzyme come, but remember that modification enzyme only work when the DNA is hemimethylated. So as here is no methylation is present, so modification enzyme do not work. So here modification enzyme do not work. Okay, so in this way the restriction modification system works. Okay, thank you for watching this video.